Hello everyone, my name is Ahmed Mustafa. I'm an assistant professor of pathology and lab medicine at University of Saskatchewan. I'm also the clinical HLA lab director in St. Paul Hospital. I'm going to present today the HLA typing methods that we are currently using in our lab. The outline of this presentation is mainly to give you an idea about what we do and how we do it and why we do it. So what is HLA? From its name, H stands for human, L stands for leukocyte, because when they initially discovered it in their experiment with their white blood cells, it's now known that it's expressed in all human nucleated cells. Antigen, because it has the ability to stimulate an immune response. HLA are markers used by the body to determine which cells belong to your body and which cells do not. In this figure, this is a kidney cells from a donor expressing a foreign HLA. The recipient T cells will recognize this foreign HLA and mount an immune response. It's more complicated than ABO system because there are many HLA markers that make a person's cell unique. Structure of the HLA. HLA is formed of two classes, HLA class 1 and HLA class 2. As you can see in the figure, HLA class 1 is formed of a big alpha polypeptide chain, alpha 1, alpha 2, and alpha 3, and a small beta 2 microglobulin. We don't do HLA typing for the beta 2 microglobulin because it's a monomorphic chain, means it's present in the same individual and have the similar expression. However, alpha chain is a polymorphic chain and it's different between individuals. The common HLA class 1 that we do HLA typing are HLA-A, HLA-B, and HLA-C. For HLA class 2, it has a different structure. It's formed of an alpha chain and a beta chain. As you can see in the figure, alpha 1 and alpha 2, and beta 1 and beta 2. Both are encoded by two different genes, and both are polymorphic except in the DR. So the three common types of HLA class 2 that we do HLA typing in the lab are HLA-DR, HLA-DQ, and HLA-DP. As I mentioned before, HLA-DR is formed of only one alpha chain, so we don't do HLA typing for the alpha chain of the DR, but it's formed of four beta chain, HLA-DR beta 1, DR beta 3, DR beta 4, and DR beta 5, and I'm going into much more details in the next slide. HLA-DQ and HLA-DP, both alpha and beta chain are polymorphic, so we do HLA typing for both the alpha chain and the beta chain. As I mentioned before in my previous slide that there are four different DR beta that we do HLA typing. They found there is a strong linkage disequilibrium between DR beta 1 and DR beta 3, 4, 5. Which means the expression of DR beta 3, 4, 5 are strongly associated to the type of the DR beta 1. And you can predict the DR beta 3, 4, 5 based on your DR beta 1 result. The DR beta 1 and the DR beta 3, 4, 5 are inherited as one haplotype, and each individual will inherit one haplotype from the mother and one haplotype from the father. For example, if someone inherited the DR1 haplotype, as you see in the figure, you can see that this individual have one DR beta 1 gene and there is no DR beta 3, 4, 5. In another example, if another individual inherited the DR51 haplotype, which is formed of the DR beta 1, 15, 16, they will have the DR beta 5 associated with this haplotype. In another example, if another individual inherited the DR52 haplotype, which consists of the DR beta 1, 03, 11, 12, 13, and 14, they found that the DR beta 3 is as strongly associated with this haplotype. In the last example, if an individual inherited the DR53 haplotype, which consists of the DR beta 1, 04, 07, 09, they found that the DR beta 4 has a very strong association with this haplotype. The first characteristic feature of HLA that I would like you to know, it's a polygeny. Poly means several and gene means genes. That means it's formed of several different genes. The HLA gene complex resides on 3 million base pair stretch on chromosome 6 short arm. This stretch contains over 200 genes, 40 of which encode for the leukocyte antigen. As you can see in the figure, HLA class 1 genes are separated from HLA class 2 genes by the HLA class 3 area. Another important feature I would like you to know is the HLA haplotypes. As I mentioned before, we inherit half of the haplotype from the dad and half of the haplotype from the mom. The HLA genes are inherited in the form of haplotypes, which is a block of genes carried in the same chromosome. So if you know the HLA typing of the parents, you can predict the HLA typing of the offspring. In the above example, you can see the father haplotypes are haplotype A and haplotype B. If you look at haplotype A, you can see that the block of genes in this haplotype is A2, B62, CW10, and DR4. If you look at the sets of haplotype in the small b, it's A31, B60, CW10, and DR4. He can pass the same haplotype as it is to his offspring, as seen in offspring 1 and 2, which carry the haplotype A, and offspring 3 and 4 carry the other haplotype from the dad. The other haplotype in these offspring come from the mom. The chance to get 2 HLA identical siblings is 1 out of 4. Another major difference between the HLA system and the ABO system is the HLA is codominant expressed. However, in ABO, only the dominant allele is expressed. In this example of an immune cell expressing both the maternal in pink and the paternal antigen in blue. So for example, you can see the HLA-A1 comes from the dad, the HLA-A2 comes from the mom. The B27 comes from the dad, and the B8 come from the mom. The CW1 come from the dad, and the CW7 come from the mom, and so on. So you can see half of the genes comes from one of your parents, and the other one comes from the other parents. Another important feature about the HLA is polymorphic. It's known to be the most polymorphic genetic system in human. This means there are thousands of alleles present for each gene. The graph shows on a yearly basis the number of HLA loci discovered every year, starting from 1987 till 2021. Every year, new alleles are added. So if you look at 2021, you will see that we have more than 21,000 new HLA class 1 allele and more than 8,000 HLA class 2 alleles. Because of these thousands of alleles, the World Health Organization had made a committee and named it the HLA Nomenclature Committee. The function of this committee was just to name new alleles and give them numbers. So in 2010, they came up with this idea that you see in the figure. First, you write the HLA, followed by a hyphen, then the gene name. The gene name could be A, as seen in this figure, or could be B, could be C, could be DR, or any of the HLA class 1 or 2 genes. And then an asterisk is added or a separator. The first 2 fields after the asterisk represent the serological typing, as this was the allele discovered by the serological method. With the introduction of molecular or DNA method, more alleles were discovered, and then they added a semicolon followed by another field with a subtype of this family. In the clinical field, we call the first field resolution typing or one field resolution typing. If you have a complete typing, we call it high resolution typing. The third field and the fourth field are not clinically significant and are not used in the HLA typing. HLA typing by serology. So first I would like to give you a brief idea about how the field evolved. The molecule of HLA was described in the 1950s independently by 2 groups of scientists. The first scientist was named John Doucet in 1952 when he was working in the lab. He noticed by mixing a glass slide which have serum of patient who has been previously transfused with leukocyte, he noticed that mixing the patient's serum with the leukocyte from another individual, it agglutinates, as you can see in the picture. He also noticed that the patient's serum agglutinates some of the leukocyte and some not, and he noticed that it never agglutinates the patient's own cells. So he hypothesized that agglutination is due to the presence of allo-antibodies that was stimulated by the transfused blood. He liked the idea and started playing with the sera from other transfused patients, and he was able to identify at this time 4 different sera. These 4 sera identified the same antigen on the cells. He named this antigen as MAC or MAC, the 3-letter based on the first name of the 3 of his volunteers, M, A, and C, respectively. Antigen MAC, later became the HLA-A2, was present in approximately 60% of the French population. Shortly after that, a young scientist named John van Roo from Holland studied multiple sera of patients who was transfused with leukocyte. He used a simple computer program that helped him to identify the alloantibodies, and he was able to identify 2 sets of series of antibody that react similar fashion, and he called them histoantigen 4A and 4B, which is now known as the BW4 and the BW6. The top right is from Van Roo computer analysis, which show the cluster analysis. This was done by an IBM computer which was owned by the Dutch Bureau of National Statistics, and computer at this time was a little bit scary. So using this computer algorithm, he was able to cross-react 8 different serums with large panel of leukocyte. So as you can see, each black dots is equal to a positive agglutination, and the dashes is equal to a negative reaction. So from this analysis, he was able to subgroup them into 2 clusters, the 4A and the 4B. And each dot is a different serum tested on a different cell. In another part of the world, Rose Pine from Stanford University was using a similar agglutination assay. She was also able to identify the same pattern in sera from women who were pregnant. In 1958, this group simultaneously published a paper which actually laid the foundation of the HLA that we are currently using. On the left side, there is a picture of Rose Pine and her group who took advantage of Van Roo computer system, but they did further development of this system using a computer that was present at Stanford at this time. They did 2x2 association table or a chi-square analysis table, and they were able to identify 2 additional antigen. This antigen was known as LA1 and LA2, which is now currently known as the HLA-1 and the HLA-2. Paul Terzaghi was a pioneer Japanese-American scientist who developed the first clinically approved test for HLA typing. This is known as the complement-dependent cytotoxicity assay. He named this plate as Terzaghi plate. This was a plate which has 72 wells, and each well have a different anti-sera in it. As shown in this figure with the pointed arrow upward, the anti-sera in this well is an anti-sera against the HLA-B27. So when we added the cells from an individual who carried the HLA-B27 antigen on the cells, the cell agglutinate and lyse, and this patient is typed as HLA-B27. On the other hand, if you look at the other arrow pointed downward, the anti-sera in that well is an anti-HLA-B40, and you can see that the same cell didn't agglutinate, means this cell don't carry the HLA-B40 antigen. So in this slides, we have more details explanation about the complement-dependent cytotoxicity assay and why it was called complement-dependent cytotoxicity. So on the left side, you can see the Terzaghi plate that I was mentioning in the earlier slide. And here we picked the 3 wells of this Terzaghi plate. And we put them on the center of this slide. And you can see that the first well has an anti-sera against the A1. The second well has an anti-sera against the A2. And the third sera has an anti-sera against A3. So the first thing that we do, we added 2000 cells to each well, and then this cells is incubated with the serum for 30 minutes, and then we add 5 microliter of rapid complement, and this is why it's called complement dependent. So the complement will activate the antibody on binding or on lysis of these cells if they bind. And then we incubate it for another 60 minutes. After the 60 minutes, we look at the cells under the microscope to see which cell lysed and which cell didn't lyse. And you can see here in the first well, which is marked as A1, the lysis happened because you can see that the cell lost the cell membrane. On the middle one, which is A2, the cell didn't lyse because the cell membrane is still intact, and as you can see, it's very tiny red line around the cells. On the third well, the cell also become lysed and died because it lost the cell membrane. So the interpretation here is that the sera was able to agglutinate the cells present in well A1 and well A3, and the interpretation here, it will be this patient will be typed as HLA A1 and HLA A3. So later on, they wanted to do some modification to the assay, so they used certain dyes to stain the cells because it was so difficult to look at under the microscope for unstained cells and look for agglutination. So they came with the idea of 2 different stains. The first is called fluorescein diacetate, and the second one is called ethidium bromide. Ethidium bromide is a red dye, and fluorescein diacetate is a green dye. So ethidium bromide, if the cell loses its cell membrane, it becomes not intact. So the ethidium bromide can enter the cell and stain the nucleus. So if the ethidium bromide enters the cells and stain the nucleus red, then you can know that this cell that you are viewing is a dead cells. However, the fluorescein diacetate doesn't enter the cells, and it only stain the cell membrane or the intact cell membrane. So if you find a green dye from the fluorescein diacetate, you know that the cells are living because the cell membrane is still intact. And then they started to count the cells. And then they came with an idea, because this was a very subjective technique, and it depends between who is reading it and tech-to-tech variation, scientist-to-scientist variation. So they came with a scoring system. So they make a range of scoring systems. So they said if it was between 0 to 10% cell death, this is negative. If it's between 11 to 20%, it's still negative. And then if it's between 21 to 50%, they call it borderline. And then if it's 51 to 80%, this is called positive. And if it's 80 to 100%, this is strong positive. And in this slide, you can see an actual chart of this serological assay. And you can see the chart is divided into different columns. The first column, the serum ID, and then the second column is the well or the location of this serum ID, the specificity of this serum, because you have to know the specificity, otherwise you wouldn't know the result of your patient. And then the fourth column is the results section, where you put your actual scoring system depend on the previous slide. Does it take 1, does it take 2, 3, 4, 6, and 8, as I mentioned. So if you look at the serum ID, this is the serum which comes from different individuals, and every serum has a specific ID. So usually the first 2 serum act as the positive and the negative control, and then starting from the third serum is our actual serum coming from the patient. So for example here, serum ID 436, which is present in the third position. This is located in well 1C, and the specificity of this serum is it acts against A1 individual. So if an A1 individual is positive, then you will find this well become positive. If an A1 is negative, then this will become negative. If you look at the fourth position, which is serum ID 568, and this is located in well 1D. You can find that the specificity of this one is AW36. And if you look at 521, which is located in the 5th position, or well 1E, the specificity of this serum is A2. And if you look at the result, you can see that the scoring system was 8, means that this patient reacted against the A2. And in the other example here is the 491, and the 491, the specificity is A26, and you can see that the result is also 8, meaning that this individual is typed as HLA A2 and HLA 26. The other wells didn't react, and it was negative. So this means that this individual is only typed as HLA A2, HLA 26. The complement-dependent lymphocytotoxicity assay, or the CDC, was the gold standard typing method until the late 80s, when the DNA-based or molecular typing method began to develop. But until now, there are still labs use the CDC for HLA typing, or at least they use it for cross-match, as we will see in our cross-matching presentation. So DNA-based typing and serological typing, there are two different entities. Serological typing, from its name, means it's an antigen-antibody reaction, means that these cells or the antibody is reacting or binding to a specific antigen present in the cell. DNA-based typing means that you're looking at the genes or the alleles expressed at the gene level or at the nuclear level. So in this example, you can see this is a cell from an individual molecularly typed as A0201 and A0203. And as you remember from my beginning of this presentation, I mentioned to you that usually you receive one haplotype from the mother, one haplotype from the father. And this patient is typed as a heterozygous, means there are two different alleles of the A02. One of them is A0201 and one of them is A0203. When these alleles form the protein is going to be expressed on the cell surface. However, this protein is going to be an A2 protein because both of them will form the same protein, but they have different genes. One of them is A201, A0203, but both of them will have the A2 protein. When you put an antibody that recognizes the A2, this antibody will bind to the A2, and this is called the serological method. So by serology, you can type this patient as an homozygous A2, means he has the same A2, but with the molecular method, this patient will be typed as a heterozygous A0201 and A0203. So the first method of HLA typing by molecular method is the sequence-specific oligomer, and we will get in much more details in the next few slides. So this is just a picture of the machine that we use in the lab for doing this method of HLA typing. There are two common machines. One of them is the Luminix 200 and the other one is called the Flex Map 3D. Both of them are a microflow cytometry. The difference between the Luminix 200 and the Flex Map 3D is the resolution of typing. So Luminix 200 can count up to 100 beads, and we will go into much details in this. But the Flex 3D can count up to 500 beads, means it can give you a higher level of resolution of typing. So this is a picture of the beads that I mentioned earlier. So what is the beads? The beads are just like a microsphere particles. It's made of plastic or could be made of metal. And each bead has a specific probe, means a part of the DNA or a sequence, a small sequence of the DNA. This small sequence of DNA is called oligomer. And this is why the name sequence-specific oligomer comes from. So these beads are when we do the assay, we combine the DNA from the individual that we want to look for the HLA typing with these beads. If the DNA is compatible with these beads or with the sequence or the oligomer present on these beads, it's going to bind. And then we put another fluorescence label that can tag this binding, and if this fluorescence bind means that this patient is typed for this the sequence present on these beads. So let's see an example of that. So here in this example we have a bead capital A and a bead small a, and each one of them has attached to it a specific probe or a specific oligomer or sequence of DNA. When we combine the patient own DNA with these beads, you can see in this specific example that the DNA only were able to combine to bead of the capital A, but was not able to combine to the bead of capital E, of the small a. And when you look at the graph on the next, on the right side, you can see that the signal for the capital A was higher than the signal of the small a. This means that this patient is expressing the capital A. And if you look at the same example as in HLA, instead of the capital A or the small a, it could be HLA A1 or HLA A2 or HLA A3 or A4 and so on. So using this method, you can able to discriminate, or you can able to know or resolve the HLA typing of this patient. And as I mentioned, there are two types. There is the 100 beads or the 500 beads. Means if you have more beads, you can get a better resolution or a better resolving of the HLA typing rather than if you have a smaller number of beads. One important feature that I would like you to know about this microsphere beads, that they are actually coated by colors. That means that each bead has a specific color. Like in this example, you can see here a set of 500 beads, and the range of color is, if you look at the top one, is orange, and if you look at the lower right corner, is dark pink, and this is actually identify the probe attached to these beads, as we will see in the next slide. Okay, so the way this Luminix assay works for HLA typing, it's the same as the flow cytometry, but it's a micro flow cytometry. So it consists of sheath fluid and lasers. So your beads, which contain your DNA sequence, pass through the sheath fluid until it reaches the interrogation point, where it can be hit by two lasers, the red and the green laser. The red laser hit the beads, and based on the color of the beads, it can identify the DNA sequence attached to the beads, because when they initially manufacture the beads, they make the software recognize that each specific color of these beads represent a specific part of the DNA. The second bead, which is the green laser, will hit you only if your DNA bind to these beads, because when the DNA bind to this bead, it's already been tagged by a fluorescence dye. So when the green laser hit, it will identify this fluorescence dye tagged to your bead, and it will be able to quantify or qualify this bead, tell you that it's a positive or negative bead. So this is the probe hit table that you see on the top of the screen. This is how the Luminix software is being programmed. So for example, it's programmed to give you the A0101 typing when probe 1 and probe 2 is positive. When probe 3 and probe 4 are positive, means that they are hit by the green laser and they identify a tag, then the typing will be A0201. If probe 5 and probe 6 are positive, mean that the green laser was able to identify a fluorescence dye in probe 5 and probe 6, the typing will be A0301. And this is just an example. It can be programmed anything you want. And if you go more into the probes, it looks more like a pattern. Like for example here, if you look at the A1101 typing, if it's positive, if probe 1 and probe 5 are positive, both of them have to be positive. If one of them is only positive, you cannot identify the typing. And in the last example here is A2301 is positive when probe number 2 and probe number 6 is positive. So let's see the bottom example to see if you understand the concept or not. So let's look at the first. If now you run a sample, and for instance, let's say you run sample 1. Sample 1 give you probe 1, probe 2, and probe 3 and probe 4 are positive. So what do you think the typing would be? Okay, I think it's going to be A0101 and A0201. Why? Because probe 1 and probe 2 is positive, which is for A0101, and probe 3 and 4 are positive, which is for A0201. That's correct. Let's see sample number 2. Sample number 2 give you probe 5 and 6 were positive. So this is an easy one. You can just spot it from this table up on the top, and it's actually A0301. Interesting. Now it's going to get harder. What about sample 3? Sample 3, you have probe 1 and 2 positive, which is A0101, and also probe 5 and 6 is positive, which is O0301. So the typing on sample 3 will be A0101 and O0301. I think there is something missing also here in sample 3, because sample 3, probe 1 and 2 is positive, which is similar to 1101 and 2301. So in that case, the software will tell you that your typing is likely is going to be A0101 and A0301, but I cannot rule out the presence of 1101 and 2301. So this is what I call it ambiguous or cannot resolve the typing. So if you have a higher number of beads, say like 500 number of beads, which is done by the 3D Luminix, it can resolve this typing. But if you have 100 beads, it will tell you this. It could be the combination of the first typing or the combination of the second typing. And let's remove the blue box to see the result. So yeah, as I mentioned, sample 1 is straightforward, sample 2 is straightforward, and when we look at the sample 3, it tells you it could be A0101 or A0301, but also it could be 1101 and 2301. If we look at the pattern in sample 4, you can notice that this pattern is a new pattern, is not present in any of the previous sample. Like for example, it could be, yeah, it matches actually 0201, but what about this extra box? Is it half 0301? So when we find a pattern like this, it could be a novel allele or could be some sort of technical error, and extra investigation has to be done. So this is the actual report that comes from the Luminix, and I would like to highlight some important points here. So for example, this is a case of HLA typing for the gene A, and on the top of the screen you can see that the typing is A02, and then there is XX1, and the other allele is A03, and then XX2. So this means that the machine is able to identify the low resolution typing, which is O2 and O3, but cannot resolve the high resolution typing. because this is the idea of the Luminix. The Luminix can only resolve low resolution typing because of the number of alleles, because of the ambiguous that it cannot resolve. So when you look at the XX1, this means that on the below the typing it says XX1, and then there is a lot of alleles here. This means that this allele could be A0201 or A0206 or A0207 and so on. And if you look at the XX2, this means that this is O301, O303 null, or 3034, or 305, and so on. Which means that the Luminix is only able to tell you the antigen level of resolution, but cannot tell you the higher resolution typing. And therefore, you need a different method to identify your high resolution typing. So, as I mentioned before, that you need a high resolution typing to resolve the ambiguous that happens from the Luminix or the SSO, which is the sequence specific oligomer. In the lab, we used more specific primer, and this technique is called sequence specific primer. And this gives you a high resolution typing, or a two-field resolution typing, because if you remember, low resolution typing is just one field. High resolution typing is two-field. So if you remember from our last slide, the Luminix were able to identify a low resolution typing, which I gave you an example of A02 and A03. Now the next test that we do, we now identify that this patient is A02 or A03. So we can now bring a tray, as you can see here on the right side of the slide. This tray is only specific for A02, so it doesn't detect any other alleles, only A02. And now because it's more specific just for A02, you can see that each well, like this maybe could be a 72 well tray, or a 96 or a 16 or a 64 wells. Each well has a specific primer that can identify a specific allele of the A02. Let's say an example. If we look at the first well could be A0201. The second well could be A0202, and the third could be A0203, and the fourth could be A0206, and so on and so on and so on. So this tray become more specific and give you allele level resolution or high level of resolution. So the way we do it, we just add our DNA, we add our primer, and we add our tag, and we do a PCR or a PCR amplification of this product, and then we run them on a gel. So in the middle of the screen, you can see there is a gray box, and then there is some lines showing up. You can see if you see a second line or a second band, they call it bands, this means that this band is positive. If you didn't see a second band, this means this band is negative. So in this example, you can see that the third and the fifth band are positive, but none of the other one are, and the other bands and the other lanes are negative. And each lane corresponds to one of the well in the tray. So well number 1 could be in the lane number 1. Well number 2 could be in lane number 2. Well number 3 could be in lane number 3. And you can see here that well number 3 is positive and well number 5 is positive, because it shows you 2 bands in lane 3 and lane 5. So from this pattern, we can identify the high resolution typing, and we will see more in the next slide. So what you see here in this slide is an actual chart that comes with a specific tray. In this example, and you can see this is for an HLA 11 tray. How did I know that this is an HLA-11 tray? Because you can see on the first column, it tells you the HLA allele, and then there is 110101, and then in the second row you can see it's 110104, and then in the third it's 110157, and then 1101, 1102, 1103, 1104, 1105. So this is specific for 1101 tray. And then on the top of this table, you can see numbers from 1 to 44, or now 1 to 45. And this is actual the wells of the tray that we use. So in this tray is for an 1101. This tray has 45 wells. And it tell you that if well 1 is positive, together with well 11 and 12 and 14, as you can see in the first row, the typing will be 1101, which is a high resolution typing. On the second row, it tells you if well number 1, well number 11, and well number 14 is positive. So in this case, it's a different pattern than in the top. The top had 1, 11, 12, 14. The second has 1, 11, and 14. Then the typing is 110104. So this is actually a very high sensitive or high resolution typing. If you look at the next example here, which is 110157, you can see that the pattern is now different. It's well number 1, well number 12, and well number 14. So you can see that the pattern is different. So you need to have the whole pattern in order to identify the high resolution typing. If you are missing one allele or one well is not positive, then you cannot identify this allele. It could be some sort of technical error or some error in the plate, and the process need to be repeated. But in most of the cases, you'll see the pattern that you are looking at. So, for example, here in another example, let's see 1106. You can see for 1106, you need to have well number 1, well number 6, well number 11, well number 12, 14, and 22 are positive. So you have to have this. All these wells are positive in order to call this well as 1106. And this is what we call it by high resolution typing. So the first method, which is the SSO, is the low resolution typing. We usually would do this first to identify the antigen level, and then based on the antigen level, we can pull the tray that we want. So if your low resolution typing or the SSO typing give you this patient is A11, then we pull the A11 tray to know the high resolution typing. Okay, I know that I went in a lot of details on what we do in the HLA lab, but let's see what you will see in the clinic. So I'm going to discuss in the next few slides what is the HLA typing report. Okay, so this is an example of the HLA typing report that you will see in the clinic. So usually you will see something called the sample date and the tested date. So the sample date means this is the when the lab has received the sample. The tested date is when the test was done. And then you will see two rows. The first row is the molecular typing that most of the lab do now is molecular typing. And then there is a serological equivalent. Serological equivalent doesn't mean that the lab has done serology to identify the HLA typing, but this is the predicted serology based on the molecular typing. So if you add the molecular typing to a computer software program, it will predict the serology or the protein level. Let's see in the first example here. So in the first example here, it's the typing for the A locus is 00101 and 00102. When we put this into the computer program, the computer program will predict that the serology equivalent will be A01. This is very easy because A0101 and A02 is it will be have the same as protein expressed on the surface. So if you remember this from the earlier slide, so here is the two genes expressed in this cell. So it's an A0101 and A0102. When they form the protein, they will form an A1 protein. Both of them will form an A1 protein. Why it's called A1 protein? Because it can be detected by an antibody that can target A1. This antibody cannot differentiate between the A0101 or the A0102. It just detects the A1 protein present on the surface. Why this is important? Because this is important to detect the donor-specific antibody. When the physician requests a donor-specific antibody, they want to see which antibodies are present against the antigen. They are looking more at the antigen level, not at the molecular level. So they want to know what is the possible antibody that will be formed here. So if this is a donor typing, and the patient will form an antibody, the patient will form an anti-A01 antibody. So this is locus A, B, and C, which is the class 1 that I already discussed. And then in the second part, this is the class 2, which consists of DR beta 1 and then DR beta 3, 4, 5, and then the DQ alpha 1, DQ alpha beta 1, DP alpha A1, and DP beta 1. And you can see on the top, this is the molecular typing, and in the bottom, this is a serological typing. In this report, the report was done by a high resolution typing. That's why you can see two fields of typing. Like you can see it's A0101 or A0102 or OA or B0801, B5501. This is what we call a high resolution typing. If you only see one field, like for instance A01 only, or B08 or B55, this is what we call low resolution typing. This is the DQ, which consists of the DQ alpha and the DQ beta, and this is the DP, which consists of the DP alpha and DP beta. If you remember from my earlier slide, we don't type for the DR alpha because it's a monomorphic. All the individuals share the same DR alpha, but we only do typing for the DQ alpha and the DP alpha. So this slide is just like a more details focusing just on class 1 on the report, and you can see we do HLA typing for A, B, and C locus by the molecular method. I just put 2 arrows here just to remind you that one of the haplotypes come from the dad and one of the haplotypes come from the mom. We don't know which one come from the dad, which one come from the mom, unless we do the parents typing. And when we do the parent typings, we can know which one come from the dad, which one come from the mom. So this is just like a speculation, which one is the dad and which one is the mom. The other thing is the serological equivalent, and I mentioned before that we don't do serology, but we put them in a computer program, and this computer program can predict the serology. If you look at the example of the A and the B, you can see that the molecular typing is very similar to the serology. But if you look at the C locus, you can see that the molecular typing was 0303, but the serological equivalent was 9. And the reason here it's because when they identify the antibodies, they identify first an antibody against the C was an anti-CW3, but then later they found that there is different subtypes of this antibody, and then when they decided into the next number was actually 9. When they found that the O303 is different from the O304 or the O301, then the next number was 9. And this is why here the serology is a little bit different from the molecular method. And this is why a computer program is needed to identify the serological equivalent. So in this case, if this is a donor typing, and then the patient can form an anti-CW9 antibody against this typing. Another reminder that we only do HLA typing for the alpha locus for the HLA class 1. We don't do for the beta 2 microglobulin because it's a monomorphic. However, the alpha gene is a polymorphic, and you need to identify which allele the patient or the donor have. For the class 2, I'm going to divide them into 2 slides. I'm going to discuss the reporting of the DR first, and then we'll discuss the DQ and the DP. So here in this slide, you can see that we do typing for DR beta 1, DR beta 3, DR beta 4, and DR beta 5. And another reminder, we don't do HLA typing for the DR alpha, just the DR beta. If you look at the serological equivalent, you can see that DR beta 101 is equivalent to the DR1, but the DR beta 0301, the serological equivalent is DR17. So it's not usually a straightforward serological equivalent. The other thing we do typing is the DR345, and if you remember the haplotypes that we were discussing earlier, and the strong association between the DR beta 1 and the DR beta 345, you can, from the DR beta 1, you can predict the DR beta 345. So in this example here, the haplotype that was inherited from the dad is DR beta 0101. And you can see that the DR beta 1 haplotype doesn't have any association of the 345. So when you see something like this in the report, you know that it's by association they are not present. It's not that we didn't do the typing. The other haplotype that was inherited from the mom, which is the DR beta 0301, you can see this is the DR52 haplotype, because DR beta 103 is associated with the DR beta 3. And then the other DR beta 4 or 5 are not associated with this specific DR beta 1. With regards the DQ alpha and the DQ beta, we do HLA typing for both the DQ alpha and the DQ beta because both chains are polymorphic, and the same also for the DP alpha and the DP beta. We do HLA typing for both chains because they are polymorphic. Thank you for listening.